Hello everyone, welcome to BrainMap. This seminar series is co-sponsored by P41 Funded Center for Method Scan Mapping housed in the Martinos Center. It is our great pleasure today to have Dr. Jing Cai for giving us BrainMap talk. Jing Cai is currently an instructor at Harvard Medical School Neurosurgery Department in Massachusetts General Hospital, where she began a research fellow in 2018. Prior to this position, she was a postdoctoral researcher in physics department at University of Pennsylvania, where she received her PhD in 2015. She has also worked at the core modeling team at JP Morgan Chase after obtaining her PhD. I'd like to remind our audience that address their question at the end of the talk. Stage is yours.
Yeah, thank you for the introduction. We're talking about a random walk model, sounds like. So yeah, it's my great pleasure to be here and meet you guys at the Martinos Center and to share with my work on the language communications. Especially we are able to use intracranial recordings with single neural recording or local field potentials. And we are also combining the large language models in this study. Okay, so human language is very complicated. So usually we are able to convey very complex the meaning through this combination of, and also in English, normally we have like actively using 170,000 words in English that people are actively using. And also it's complex in the sense that we have to assemble words in certain order so that other people can understand. And the last feature is that usually speaking is very fast, dynamical tempo. So usually we are conveying around 150 words per minute, and other people usually have no trouble in understanding us. And this is quite a lot of computation happening in our brain. Previous work using fMRI has identified the brain areas that are supporting language comprehension specifically. So overall, we found there's interconnected brain regions, including the temporal cortex and the prefrontal cortex. But however, we don't really know much about how language is conveyed in the single neuron level. Where specifically the first part of my talk is to asking how is the meaning of a specific word represented at the single cell level. So to do that, we are utilizing the very rare opportunity to record single neurons during craniotomy. So we are able to put in channels, electrodes into the posterior middle frontal gyrus, and then these contacts or electrodes are able to record the voltages from the nearby regions. So this is one example of showing one channel of voltage recording. So as you can see that there are some spikes or signatures of the individual neurons. So if we zoom in a little bit, you can see there's like one spike, another potential spikes, and they may differ by like the shapes, the amplitude, or the durations of the spike, so which may potentially indicate that they may be coming from different neurons. So in this way, so for example, there may be two potential neurons that having different like action potential features, so we are able to tell apart, so this is neuron 1, this is neuron 2. So in this way, we record single neurons from this region of the brain, and so that we can actually having the action potentials of the neurons by time. So that's the recording part. And then in the meantime, we asked the participants to listen to natural sentences, such as the child bent down to smell the rose. So these are like designed sentences that have specific, very different meanings into the 8-word sentences. And in the meantime, we have the audio recordings of these words. And we also have the neural recording. So we have these action potentials. We know when the neuron will spike. And so we can align these audios with the neural activity. So specifically, we are interested in, so right after the onset of the word, how many times do the neurons spike? So in this way, we can calculate the rate, or like say within, for example, 400 milliseconds, how many times these neurons spike, and then we can calculate, okay, that neuron spikes for like 2 hertz or something like that. So in this way, we have our data is more like single neurons. We calculate the firing rates, and then we align the firing rate to the onset of the word. So we know the neural activities after the word onset. Okay, so these are the data, and we also want to find a way to represent the meaning of the word. So we use a pre-trained model called word2vec, and this model is trained so that the embeddings are capturing the meaning of each individual word, which is called the lexical semantics. So specifically, each word is represented by a 300-dimension embeddings, and these embeddings has the character that say words with similar meanings will be next to each other, say like warm and sunny, whereas words with similar meanings are next to each other like a son and a sister, but if they differ in meanings, they are represented with a further distance. So because of this, we are able to run a spherical k-mean clustering to categorize these words into different categories. So specifically, we represented each individual words with the 300 dimensions, and then we run the clustering method so that we turn into these individual 300 dimensions into 9 different clusters. So this is unsupervised learning, but they automatically just group words with similar meaning, such as warm, sunny, cloudy, it's all like nature, versus bunny, lizard, horses, they are like animal clusters. So here we are changing the individual word meanings by these lexical semantic domains that are representing the clusters of this word meaning. Okay, so now we can directly asking, do these neurons carry the information of these semantic categories? So here we are showing 2 examples from 2 neurons. So the first neuron is neuron 15 that whenever it hears the word in the family domain, it changes the activity. So the x-axis, in here, each dot represents a spike or the action potential that the neuron fires. And then each row labels a word that belongs to this category. And then, as you can see, right after the onset of the word, the action potentials get much higher compared to the other places. We also compare to the other words that are not in the category. So this shows that just seeing these raw raster plots, these neurons have increased the firing rate after the word onset, compared to like if it hear the words from other categories, it doesn't change the activity much. So these are indicating that these neurons are encoding specifically to the meaning of the word in this category. Okay, and similarly, we are able to find another example of another neuron that are encoding to the emotion clusters. So it changes activities word for another specific lexical semantic domain. So putting together all the neurons we recorded, we found there are 14% of these neurons that display the responses, which is significantly selective to the specific semantic domain. Okay, and then we are also able to ask whether, say, each individual neurons are preferred to encode one specific domain, or they are possibly encoding multiple of these domains. So we put a bivariant chart in here, but overall we still see like a lot of the neurons are encoding only one specific semantic domain. So just by the histogram, we see like majority of them are selecting to only one domains. So this shows that potentially they are kind of tuned to the specific information of the lexical semantic information they carry. Okay. And then based on this, we can also ask, say, now we have this population of the neurons that are, say, composed of individual neurons. So can we put them together and then ask whether we can decode the semantic meaning, the meaning of the word, using all this neural population? So we perform a decoding analysis with the firing rates for each neuron and for each word as our features to predict the specific meaning clusters of the word. So this is our results as an example that we are able to actually predict pretty well in the sense that, so for this sentence, the probability of the corrected category is mostly higher than 50%. So it means that we are able to predict for this sentence a lot of the words or all the words correctly in here. So overall, through like all the words and neurons putting together, we are able to predict, decode the lexicosemantic at accuracy of 31%, which is in here. And this is compared to around 11% chance level. So overall, they are definitely having the power to decode the meaning of the word. And especially these decoding models are run on the words that have not been heard in the training data. So this is we separate the training and testing data on the word level. And also we see that with more neurons included in this model, they are able to have a higher prediction decoding accuracy. It means each individual neuron may encode one specific information, and the more you're adding in, they come up together like a population to decode, to represent the meaning of the word. Okay, so the last part is that we are also asking, because these language material are given as the auditory input of auditory sentences. We're asking whether these neurons we identified are responding to the pronunciation of the word or not. So we use homophones, which are words that pronounce the same but have very different meanings, such as sun versus sun. So one belongs to the family cluster, the other is the nature cluster. So we ask, say, do these neurons change the activity higher with homophones compared to other words that in the same cluster? So we have the words with same pronunciation but different meanings versus words have similar meanings but different pronunciations. So we see in here that these homophones, neurons responding to the homophones with a much higher differences compared to the words with the same meaning. So which indicates that these neurons are more carrying the semantic aspects of the language instead of the pronunciation. Okay, so this is the summary of the first part of my talk, is that we are able to track the individual neural activities, and we found that they are directly encode the lexicosemantic information of specific word. And this encoding is independent of the phonetic form. Okay, questions?
Is there any differences related to the timing? If I recall from the figure,
Yeah.
was 2 or 300 milliseconds after the word. Were there differences that you saw in terms of time for different categories of words?
Yes. So the timing-wise is that we see usually the peak is around, depending on how fast the language material. So usually, so in this study, we use 200 to 500 milliseconds window after word onset. But on the other analysis, we may use another window. If it's faster, right, like normal speaking, it may be 100 to 400 milliseconds. But usually there is a 100 milliseconds delay to get the signal process. So we didn't really examine like by different clusters whether the timing changes. Yeah. Mm-hmm.
This analysis was conducted in a single subject or the group of subjects?
So these are combined from 13 subjects. So each individuals we recorded, like we have 5 channels, 5 electrodes, and we recorded like dozens of channels, neurons, and then we concatenate them.
So the technique is kind of robust.
Yeah. The neural sorting and everything, the techniques is yes. Mm-hmm. Yeah.
Do you observe any improvement if you use more than one neuron for the decoding analysis?
Yeah. So the more neurons we add, so we are, for in the decoding analysis, we only use the significant neurons, and the more neurons we adding in, the higher the decoding accuracy we get. Yeah, the totally we can reach to 30%. Yeah. Or do you mind? Yeah. Yeah, sounds good. Yes, and so now I was focusing on language comprehension before on the single neuron, but we can move one step forward into the language production and then asking whether we can still like decoding the lexicosemantic meaning during language production. So this is serving as the brain-computer interface in the sense that, say, people may have trouble in speaking, so they are losing the power to communicate. So if we can decode like from their brain activities, directly translate what the words they are going to say, then this can help people to regain their social communicative skills. So, okay, so previous brain-computer interface studies majorly focusing on using the pronunciation or the waveforms of the languages that they spoke. So specifically, for example, that for Eddie Chang's paper, and they are able to recreate the waveform of the language production at a very good accuracy. However, there are very little work focused specifically on the lexicosemantic decoding, or the word meaning decoding. So as a result, we don't really know how well we can decode the lexicosemantic information. And also, but lexicosemantic decoding may potentially allow faster speed to translate the neural signal into words, and may also potentially has the benefit of if there is a low signal quality, then it may help into directly reading the meaning of the words instead of go through the pronunciation. Okay, another aspect we want to address is in the natural setup. So a lot of experiments are, say, given the blocked design saying, can you read this sentence? But in reality, what we want to restore is in the natural setup, in the natural conversation. So can we decode the meaning that the participants speak, say, when they are having a normal conversation? So in this concept. So in here, we are conducting a one-to-one natural conversation with the participants. So they are epilepsy patients who are undergoing their epileptic monitoring process, and we record the local field potentials with depth electrode. So overall, we recorded 14 participants, and so with, say, the participant and the experimenter one-to-one conversation, and the experimenter spoke around 50,000 words. And we are able to get the participants engaged in the conversation so that they actually spoke around 40,000 words. So in here, specifically for this purpose, we are focusing only on the words spoken, on the language production part of the conversation. So in the meantime, we are recording the intracranial stereo EEG recording. So these are depth electrodes that are locating on different brain areas, so majorly focusing on the temporal lobe and the frontal lobe. And then what we record are still the voltages in the channels, and then from the voltage, we are using Fourier transform to get different frequency bands, and then calculate the envelope of each frequency bands. So different frequency bands may indicate different brain circuits or different level of circuits. And now we can also align the onset of the word with the frequencies. So similarly, how we represent the lexicosemantic information, we are still using the word-to-vec model. And then we do exactly the same, embed each word into the 300-dimension embeddings, and then do the k-means clustering, spherical k-means. So similarly, we see words with similar meanings are grouped together, and this is more like the family clusters, and then some university stuff. It's also the results are overall making sense. So now we can do the decoding. So the methodology is similar, but now we are using the channels and the frequency bands as our features for each word. And then we using these features to predict the semantic domain. So we now cluster them into 10 different domains. So we run a logistic regression model in here, and then across the 14 participants we recorded, we found an average of 21% of the decoding accuracy. And this is compared to 10% of chance level because we have 10 clusters. So overall, this is showing that the whole analysis in here is that we demonstrate the feasibility of lexicosemantic decoding from the natural speech that is not a blocked design. Okay, and then, so we can also further ask what part in the brain where that is specifically supporting this lexicosemantic production planning period. So we're asking which channels are responding to the lexicosemantic cluster domains. So we found there are 12% of channels across all the participants that are specifically responding to the lexicosemantic clusters. And then across different frequency band, we do see there's a strong bias towards the lower frequency, such as theta or alpha band. So these are both, like there are higher percentage of the selected channels in the lower frequency, but also when we put into the model, these lower frequency band are having a higher feature importance. So they're adding more accuracy toward to the end of the accuracy compared to like higher frequency bands. So this is the plot showing where the channels are contributing to the lexical-semantic domain decoding, but we can see is still like widespread into different brain areas. So what we tried to decipher it is to ask, say, given one specific areas, can ask whether the channels in the area are showing a significantly higher ratio than channels from the other area. So in this way, we can identify 3 regions that are having more significant channels than the other areas. So these are middle temporal cortex, precentral, and the pars triangulus. So in these 3 areas, showing in here, like we are able to find a higher percentage of channels that are responding to the meaning of the word. So now we can ask, so what if that I only take these 3 areas channels and then put into the model? Like, can these 3 areas encode already all the information I need to decode the lexical semantics, or I need channels from other areas? So running the semantic decoding model from the channels only from these 3 areas, I got 17.4% of accuracy compared to like if I use all the channels together is 21%. So it is significantly higher for sure than chance, but it's still not as high as using like all the channels from all the areas. So this includes that, this like suggests that the channels from other areas are still contributing to the lexical semantic processing. Okay, so to sum up for the second part of my talk, so we are moving one step towards the brain-computer interface side, which is using the lexical semantic decoding model and ask whether we can use intracranial SEEG recordings to decode the meaning of the word during language production. And we do see like it is possible. We got a much higher percentage than chance. And also we found that the neurons are the channels are widely distributed across different brain areas, even though they focus on the 3 regions more. And the lower frequency band are showing a higher response to the lexical semantics. So I will take 1 or 2 questions or in this session. Yes. Mm-hmm.
So this is a scalp EEG, right?
No, it's intracranial. Yeah.
Okay. So that's the reason why you are running this on epilepsy patients, right?
Correct, exactly. Yeah, yeah.
Does the fact that they are like epilepsy patients like have an impact, do you think, on this type of results, or?
Just say that again.
Does the fact that they are like epilepsy patient has any impact on those areas? Like it's like something inter-subject that you have to take into account for those patients?
Yeah, so they all have intact language ability, like conversation are normal. And secondly, we do see these epileptic discharges occasionally in the channels. So we detect like how many of these charges happening, and if the channel have more than 6 spikes per minute or something, we just remove the channels. So yeah. Mm-hmm.
One quick question. Do you have intracortical or microelectrodes or any way to look at the different cortical layers in these reports?
The channel itself are like millimeters in size. So
this is just regular SEEG?
These are regular SEEG, yeah. Yeah. All right, so we can then, yeah, go ahead.
So in terms of like the different frequency bands, can you speculate why certain frequency bands are decoding better than the others?
So this is just my interpretation, which my understanding is that lower frequencies are more from like top-down projections of expectation stuff, whereas the lower frequency bands higher frequency, like gamma bands, are more correlated to the sensory part, like bottom up. So if I listen to stuff, then it will be like more sensory coming up versus if I want to speak more, it will be more top-down projection. So this is my interpretation, is like the lower frequency may be correlating more like the top-down planning phase than the sensory like checking errors thing. But it's a little like, it's my interpretation. It's hard to justify that. So now we can also, like now we already have the natural conversation data. So why don't we study natural conversation? So again, natural conversation is more complex than language itself. So not only we exchange language into like convey unlimited information, it's also highly dynamical, and the information is unbounded. It's like I can say whatever I want. It doesn't have to be anything real, like a pink elephant or something. And there are different components of the natural conversation, such as language production, language comprehension, and then the speaker may take turns between the speakers. So if you interrupt me, then you are starting to talk, and I start to listen. So as a result, previous work are trying to use the block design paradigm to study each part of the conversation, such as, say, the language pronunciation, lexical, semantic information, and so on. And then using this way, we are able to identify the brain regions that are encoding for language comprehension and production specifically. But these are all reductionist approaches, so we don't really know like how well these findings can still be applied into natural conversation, and then they don't really treat natural conversation as a continual process. Instead, they chop into trials. So specifically, there are a couple of questions that are not answered, such as how is the neural process of contact represented in the brain? And are there common neural processes involved in language pronunciation, language production and comprehension in the conversation? And then we also don't know what happened during the transition, speaker transitions, and whether the turn taking is related to the information that get conveyed during the dialogue. Okay, so to address that, again, it's the same data set that we have experimenter talking to the participant. So now we're coming back to the challenge. It's like natural conversation is unlimited potentially. So how can we find a way to represent this high level of linguistic information that exchanged? So similar in here is like similar to word-to-vec model that represent lexical semantics. Can we find another model that are showing representation of the language itself as a compositional feature or representation? So yeah, we turn into the large language model because they have very good performance overall. And yeah, so there are quite a lot of development recently. And so we use the pre-trained large language model. So usually they have the features or the structure is like layered structure. So language are represented as, so each individual words are represented as a vector and input into the input layer. And then through complicated processing, this information got processed and carried by the first hidden layer. So the whole structure got duplicated multiple times until you reach the top layer, where this layer is directly connecting to the output layer, is what your model is training on. So for example, okay, I'm using a GPT-2 model, which is the great-great-great-grandpa of the ChatGPT model. So this model, the input is part of the sentence, and then it's trying to predict the upcoming words based on the previous part of the sentence. And this sentence is carried out by 12 of the modules that have very identical structures in it. And between these modules, information are embedded into these GPT-2 neurons. They have 768 dimensions of these. So we can get these artificial neural activities for each of these words. So this forms the counterpart of how our brain reflects the ongoing of the words. So previous work is showing that the lower level of these models are representing more of the word level information, whereas the higher layers and levels are representing the integrated like sentence or phrase level information. Okay, so now we have our neural activities and we can align them into the outside of the word. And we also have the GPT-2 model. So we can use the transcribed word that both the participants speak and listen and put them into the GPT-2 model. And then we can get the GPT-2 embedding. So this serves as the counterpart of the is more like the artificial neurons that are embedding on the same words that the actual conversation has been like spoke and listened. So if we can see, and then we can correlate these artificial neural activities to the channel activities. So if they show significantly correlation, it probably indicates that the neural activity in our brain is encoding language itself. So in other, we are using these GPT-2 or large language models as a way to represent language. And they form a vectorized representation, and then we correlate to the neural activity. So any neural activity showing significant correlation is highly possible processing language. So when we say correlate, there are two examples. So in here, it is showing that the neurons are significantly correlated to the embeddings of the GPT-2 model. So the purple line is showing the GPT-2 model, and then the blue line is showing one example of one channel activities. And as a comparison, the figure on the right is showing an example that is not correlated. So in this way, we can identify the channels that are significantly correlated to the GPT-2 model. So we can plot these channels in here. And so they are, again, widely distributed across different brain areas for both language production and comprehension. So if we group them by the brain areas, we are also seeing that these are overall, like quite a few of the areas are showing higher than chance performance, significant percentage of the significant channels. So for example, in the precentral area, we see a significantly higher ratio during language production, whereas say middle temporal cortex, superior temporal cortex, or even hippocampus are showing quite some overall significance of those channels. So we also ask, say, because we were using the old version of all the large language models, so what happened if we turn into a newer models, such as, say, a FICON model or some of the like Llama model that got much higher performance in language, say, tasks. We still find that overall they do show significant percentage of the channels that are encoding, that are correlated to these models. But the interesting part is that when we arrange these models based on the size, so x-axis is increasing of the size of the model, we do see a higher and higher percentage of significant channels. So which means that the larger the model, it usually have a higher percentage of correlated channels. So it implies that the larger model can explain the neural activities better than the smaller models. Okay, so what about different frequency bands? So in here, we see like a slightly middle gamma frequency band are showing higher for the language comprehension. So for language production is overall similar, even though mid gamma still show a little bit higher. But these are like average across all the brain areas we recorded. And then we can ask, so now we have the same representation of large language model for both language production and comprehension. So we can directly asking whether the same channel responds to both production and comprehension together. So we see overall there are quite like 80 of the channels, significant channels that are responding to both modality. However, it is significantly higher than chance, but compared to like there are still more channels responding to only one modality. So we got a partial overlapping of these channels that are responding to both activities. So yeah, and these channels are also like distributed across different areas. So now we can ask what happened during transitions, like I speak versus you speaking. So how the neurons are encoding these turn-taking activities. So we directly asking, say, during transition, whether a channel's activity significantly change during transition versus not during not transition, during established conversation. And we found around, say, 15% of the neural activities that are specifically tracking the changing of the speakers. So this analysis is totally like just the raw analysis on the transitions, and it got nothing to do with the large language models. But we do see that they show quite some overlapping between these transition channels and the large language model correlated channels. So which means that, say, when people interrupted me or when I start talking, then my neurons, my channels that are responding to the transition of the speakers is not changing their activities, not just because they are tracking the speaker, but they are actively making planning for the words that I'm going to speak or trying to comprehend the words that I hear. So that's why they are showing these overlapping behaviors in here. All right, so this is the summary of the third part of my talk, is that now we expanding our view to natural conversation, and we used large language model to encode the specific language activities. So we found that there are quite some brain areas are correlated to the large language model. And they also span on multiple frequency bands, even though like middle gamma band are showing slightly higher. And then we also see that the channel responding to language production is partially overlapping to those during the language comprehension. And then during transitions, we see the channels responding to transitions overlap to the responding to the comprehension and the production. Okay, questions?
Yeah. Do you find that there's a trend towards strongest correlations across the layers of these models? Are they more strongly correlating towards the output layers or the middle layers?
Yeah.
Is there any correlation of that layer behavior with the frequencies of the output?
Yeah, so firstly, especially for language comprehension, we do see like the middle high, the layer is showing like there's a little peak at around layer 8 or something. So it's going up and then coming down a little bit to the output layer. So the peak shows up around layer 8 or 9. But overall, the higher layers are showing higher correlation channels than the lower layers. And for language production, it's more flat than comprehension, but still towards the middle high layers. Yeah. We did check the frequency dependence, but I don't remember the results. Probably is not super clear on like whether there's a significant changes. But and also there's a dependency on the brain areas. So middle temporal cortex versus like other regions, such as say hippocampus or something, they don't show very similar trending in there. Yeah.
So I don't know if I missed this information from the breaks you said that this is a naturalistic conversation.
Yeah.
So what are they talking about? Like is like two friends, like is a patient and a clinician is.
Yeah. So usually the experimenter are like our people. So we just talking about normal activities like friends, families, or movies, and then like outdoor activities, hobbies, and like. What others? It's like overall like normal topic that are people chatting about or yeah.
And is depending on the conversation, is there maybe some type of emotional involvement that can change the way they process the language?
I don't know. Short answer is we didn't really dive into it. Yeah.
Okay.
Does the correlation differ depending on different subjects? Like the signal you acquire from one people versus another?
Yeah.
How does the correlation changes across different subjects?
Short answer, yes, it does depend on the subject. But there's another individual variance is that the location of the channels are very different for each individuals. So when we put in channels, the design is to monitoring their epileptic activity. So it's clinical design for each individuals to track where it is. So it's a little hard to say whether the difference is due to different locations of the channels or is like different people are different. Yeah.
Or in your understanding, like would it be like or so because of the individual differences versus the location?
We don't really know. We don't. It's a little hard because even the same location, like say superior temporal cortex, is still large. Like we don't really put exactly the same locations for different people. Yeah.
I have a question about the language model. So you are talking about model neurons. What is that the language model is actually using when it tries to interpret our speech? Is it like simulating LFPs or firing, or where do those time courses come from?
Yeah. So the language models that I use, the inputs are individual words.
Yeah.
Say I transcribe the words from the talking. So they don't really have time. But the neural activities are time series. So I need to align them. So one way is to align the neural activities to the onset of each word. So I average, so in this case, I average from 500 milliseconds window. Say if the onset of the first word is 1 second, on the time stamp 1 second, then I use 1 to 1.5 second window and average neural activities there as the comprehension. But for language production, I use 500 milliseconds before the onset of the word. So it's more like the production planning. So I align the time series to the word onset so that I have the same number of words for the neural activity. Yeah.
So two quick questions. Great job. I was just going to ask you, are all these words, the lexical semantics, looked at in the context of sentences?
Yes.
So it's not like if you had a string of words?
Correct. So we do have a control for that, which is just putting into the list of words that do not have coherent sentence. And any of the neurons showing selectivity for the specific lexical semantics are not actually responding to the list of words. So yeah, so it is depending on the context.
So you have these clusters of words that
Yes.
similarly activate the neurons depending on the lexical entity or meaning, right?
Yes.
And so then in that case, the second question I have is about the relationship between production and comprehension. Then you said that there is partial overlap, but then you also have a bunch of neurons that are different. Do you see any pattern about those ones that are different, that don't overlap? How do they relate to production, comprehension?
We don't know. Short answer is we don't know. So we are tracking for these are tracking the local field potentials, and we ask for that channel, does it select to both language comprehension and production, or does it correlate to the GPT-2 model for both comprehension and production? And we didn't. Yeah, short answer is I don't know. We didn't check like what information these channels are processing for each modality.
So the ones, from an anatomical perspective, the ones that are in the non-overlapping, do they provide insights about what might be going on that's different?
We don't know. Short answer is we don't know, but
The green one, like the ball.
yeah. So yeah, these are overlapping channels, and these are non-overlapping. So I'm plotting the overlapping channels with the bigger dots, whereas the smaller dots are for each individual modality that's not overlapping. So it's not, or short answer is by location or frequency and stuff, there is no clear, just very sharp differences. So I did dive into like checking whether there are pattern or something, but it's not very obvious for sure. And it does like distributing on all different areas we recorded.
That figure on the right does not show the anatomical distribution of that green.
Oh, sorry, the colors.
That's anatomy.
Yeah, the colors are different theme. Yeah, thank you. Yeah, sorry. Yeah. Okay.
Jesse.
Yeah.
So because of these are the semantic word input, what are the inter-patient variability? Because I believe their channels would fire differently in people.
You are asking.
With different word inputs, like some people pay more attention or perception to specific semantic lead the others.
Yeah. So for the single neuron part, because for that, the very first part of my talk, because we use the same language materials. Yeah, okay, short answer is I don't know, because each individual, we may only record like, say, a dozen or something like 10 or 20 neurons, and they are so sparse. Like out of these neurons, maybe like 5 of them have significant correlation to the lexicosemantic domain. So the overall, yeah, because we totally, for that recording, we totally have 13 participants, and we recorded 140 neurons, something like 150 neurons for this data set. So each individual participant are not having a lot of neurons to analyze the variance. And we also have another control for this analysis, which is using NeuroPixel, that we record 3 participants and a total of 150 neurons. So that gives us some power. Yeah, but overall, it's a little hard to tell the individual aspects. Yeah. Okay, so the last part of my talk, yeah, I still have time, is very short, but I want to share like our ongoing research, which like even zooming out more to talk about like social communication, because in very rare opportunity, we are able to like record 2 subjects at the same time, so we can ask them like, can you talk to each other? Like, so this is a Zoom talking, so we can record for both 2 brains in the conversation. So in theory, we have all these informations that they are sharing. And so in here, we are specifically targeting on the, say, neural synchronization, which is, say, in order to have a smooth conversation, it requires that not only I understand, I interpret what other people is saying as well as the social cue, but also I'm sending out my social information to the other person for interpretation. So as a result, previous work has shown that there is an interpersonal neural synchronization because they are able to talk to each other and interpret different aspects of the information they exchanged. So we want to study using intracranial EEG, SEEG recordings to see whether we can decipher the dynamics or specific brain areas that are encoding this synchronization. So we successfully recorded 5 participants, and we are able to correlate the pairs of the interpersonal channels. So this is just showing one example from left superior temporal cortex for both participants. So we have 5 pairs, and then each of the subplot is showing the one channel, one frequency, whether it's alpha or gamma. So as for the diagonal, we see like for the diagonal ones, we all see like at time 0, when they aligned to each other, there is quite some correlations happening between them. Whereas it's actually more complicated because if it's off diagonal, we do see like for different frequency bands, it may have a negative correlation between them. So this indicates potentially there are quite some neural integration signal happening in here. All right, so the summary of my whole talk is that, so I started from the single word meaning, and we examined the single neuron activities. We found that the single neurons are directly encoding the meaning of the word in the language comprehension. And then we transitioned to the language production, and then we ask whether we can decode the lexicosemantic meanings using SEEG, like local field potentials. And the answer is yes. We demonstrated that it is feasible to decode the word meanings in the natural conversation. And then we can ask what happened in the natural language production and comprehension. How do neural activities supporting such behavior? So we do see, using large language models, we do see like the neurons are similar in a similar way to represent the production and comprehension as the large language model. So they do show quite some overlapping activities. So then we come into the natural conversation that we can compare the language production and comprehension. And we see that they show overlapping, partial overlapping between the channels. And also these overlapping between transitions and language activities are also observed. So finally, in our ongoing research, we are trying to study the hyperscanning towards the social communication and see whether we can see some synchronization between the two brains. And these are like collaborations from the Ziv Williams lab and Sydney Cash lab. And also we acknowledge the fundings that we receive. All right, yeah, so questions?